Hello class, good afternoon. Today's class is dedicated to learning about gene therapy. And before going into this chapter, like my all classes, I'm going to go into a little bit basic concepts, and then I return back to the major slides. Okay, so as I mentioned to you, the title of this or this chapter on gene therapy. So now the question starts: why gene therapy? So gene therapy it emerges from essentially the diseases that occurs from the change in the sequence of nucleotides. So you know that the dictionary, the alphabets that constitute DNA, our genetic material, is made up of 4 letters: A, T, G, C, and our DNA is meters long, but it is made up of these 4 alphabets: A, T, G, C. Now these alphabets A, T, G, C are arranged in a fashion that ATGC, it is arranged in a fashion that it communicates, or it transcribes and translates into much bigger information, although there are only 4 alphabets, right? ATGC in DNA. 4 alphabets mean 4 nucleotides. But these 4 nucleotides, they are responsible for making million copies of RNA and million copies of proteins. And these proteins serve as a workforce to mediate various cellular functions. And this relationship between DNA and protein and RNA is defined by a concept called as central dogma of molecular biology, right? Which is that DNA, it transcribes into RNA, and then RNA translates into proteins, right? Um, but we know more because there has been advancement in sequencing technology, the ability of ours to scientists to understand the function of proteins. So DNA is secured in the nucleus, right? But DNA sitting in the nucleus sends its information through RNA, and this RNA then it translates into proteins. And proteins, they do the job of the DNA. Now, this is called as central dogma of molecular biology. Now, DNA, as I mentioned to you, it has 4 alphabets, right? Even RNA has 4 alphabets, AUGC instead of ATGC, but there are 20 amino acids that come out from RNA. Now, these 20 amino acids can form millions of proteins, and these proteins can differ in their properties. They could differ in the number of amino acid, they could differ the number of amino acid, sequence of amino acid, the chemical properties of amino acid. So protein creates lot of room for DNA to execute its function, right? So if the DNA goes wrong, what do I mean by DNA goes wrong? DNA goes wrong means the sequence of nucleotides in the DNA is altered. If the sequence of DNA in the sequence of nucleotides in the DNA is altered, it is definitely going to affect RNA and of course proteins. So all these proteins that are coming out from the altered sequence of DNA will not function as it is supposed to function. And that will give rise to diseases, right? So now how to correct the sequence of, so how to rectify, how to rectify the altered sequence in DNA that is causing disease, right? So we have to, so when you write something on the Microsoft Word, you can go back and do proofread and do spell check, do grammar check. That's not possible in the DNA if the nucleotide is already changed. So we have to develop technology, right? Technology is the only hope by which you can restore the normal DNA. So the way we can restore normal DNA sequence. Why do you want to restore? Because the altered sequence has given rise to many, it can give rise to diseases which can be passed down from one generation to the next generation. And for that generation, it could be debilitating, right? So how to fix the altered sequence, right? So now there is a new term that is being coined. It is also called as now, because there are many technologies now to correct gene disorders. Gene disorder means alteration in the sequence of nucleotides in the DNA. So these techniques are these days are called as gene editing, just like we edit Microsoft document or PowerPoint. These strategies, these technology which rectify, which fix the problem in the sequence of nucleotide to make a disease individual into a normal individual is gene editing. And traditionally, this has been called as gene therapy because because there is defect in the gene, right? Now, just want to spend some time. There's a lot of confusion with the word gene, right? So a gene could be a segment of DNA, several pieces of nucleotides. A gene could be that segment of DNA that code for proteins. A gene could be that sequence of nucleotides that could code for RNA, and RNA and protein. So for sake of understanding, genes could be referred to that sequence of nucleotides that have functional effects, right? Because if it becomes from functional to non-functional, then it will have consequences on a human being particularly. So to treat the altered sequence of DNA, to restore normalcy, normal physiological function, to treat a diseased individual, this approach is called as gene therapy. So one of the you know most known example for gene therapy is that I mostly give is sickle cell anemia. And what you see in sickle cell anemia is that in normal cell, the normal DNA would read as GAG CTC, but there is a mutation that occurs in the GAG sequence. The GAG is altered to GTG, and the corresponding complementary base is CAC, that instead of coding for glutamic acid, codes for valine. So glutamic acid is a negatively charged amino acid, whereas valine is a non-charged hydrophobic amino acid. So if glutamic acid is present, the hemoglobin molecule forms a tetramer. But if valine is present, then it starts to make, different stoichiometry of hemoglobin molecules, and which really crushes the normal function of RBC. And RBC, instead of remaining spherical, it becomes sickle shaped, right? So how to fix this? How to correct this? How to put A here and remove T, right? So This strategy is called as gene therapy. So gene therapy is a treatment, essentially, that consists of introducing into a patient a normal piece of DNA which could overcome the mutant DNA. I repeat, introduce normal DNA into a disease cell, and that cell became diseased because of altered sequence of DNA. So when this normal DNA is injected into a disease cell, the cell accepts this normal DNA and try to replace the diseased DNA by normal DNA. So there are multiple steps here, right? First of all, we have to know which nucleotide sequence is altered that is giving rise to disease. So you need DNA sequencing technology that we discussed in first or second chapter, how to sequence DNA. Once you recognize the mutation, then you have to design artificial DNA and a system that will take that normal artificial DNA into the cell. So that's number two. Then when scientists have already delivered this normal DNA, that should function inside the cells, right? Because the cell membrane is very complicated molecule, a very complicated boundary. So inserting something or injecting something foreign is challenging. So there are numerous steps in the process of gene therapy. It's a dream technology to treat diseases because there are many mutations occurring because of many reasons, right? But this chapter is dedicated to a technology called as gene therapy that can fix the abnormality in the DNA. And one of the examples I gave you was sickle cell anemia, that in which one single base change has such a tremendous effect on the RBC functions and structure. And similarly in severe combined immunodeficiency diseases. So in severe combined SCID, here it is given, it results from a deficiency or mutation in enzyme called as adenosine deaminase. If this enzyme is mutated, then these individuals cannot function to combat different infection systems. So they become severely compromised, right? So adenosine deaminase is an enzyme that participates in nucleotide biosynthesis, right? If this enzyme is mutated, then individuals who have this mutation in the immune cells in these individuals may not divide. Immune cells need to divide to combat infection. If the immune cells are not dividing, then ultimately the pathogen, the disease organism, tries to dominate over the individual, and that causes diseases. Okay? So as I mentioned to you, it's very important that every step is very thorough for the development of gene therapy. So as I mentioned to you, that one of the strategies would be to take out bone marrow cells from the patient. So let's say this patient has, sickle cell anemia. Sickle cell anemia is a disease of hemoglobin, and that is because the gene for hemoglobin is mutated. And we all know that RBCs are made up initially in the bone marrow. Bone marrow are the mother cells that give rise to RBC. They are the multipotent cells that give rise to RBC. So if we take out the mother cell, the bone marrow cells, and grow it in the lab, right? And we provide all the nutrition and conditions for the bone marrow cells to survive in the artificial condition, right? So bone marrow, it's more comfortable inside our body, but in the lab condition, it is very extremely difficult to grow bone marrow cells. And growing is challenging, plus to maintain aseptic condition, right? Like there's no contamination. So these bone marrow cells are drawn from the patient. It is treated in the lab. If the cancer is emerging from bone marrow, then bone marrow cells could be treated with chemotherapy or treated with gene therapy, and then then when it is confirmed that these bone marrow cells can now can behave normal, then it can be injected back to the patient. So what is it's definitely challenging, but what is good thing about this is that there is less chance of rejection. So if somebody gets bone marrow from a different individual, there are high chances that the person who is receiving the bone marrow can reject the foreign cells. So if we correct the cells in the patient, it is more advantageous because it closes the room for rejection. Now, there can be several kinds of gene therapy, right? Number one, gene therapy. Gene therapy could be of somatic cells, right? They're at a very higher level or broad level. There are two kinds of cells in our body. One is somatic cells, and the other is germ cells. Germ cells, they're mainly responsible for giving rise to gametes. And somatic cells, all the parts of the body except the germ cells. And we can imagine that a mutation in the germ cell will directly lead to passing on the mutant gene to new generation, right? And in somatic cells, it can divide and divide, and it can cause cancer. So both are equally and challenging to deal with. So there, somatic cell therapy, there can be two types, autologous and allogenic. Autologous means, as I mentioned earlier, we take out the cells from the patient, treat it in the lab. It could be by gene therapy, it could be by chemotherapy. But since we are talking about gene therapy in this lecture, it's by gene therapy. Then the same cell from the lab, after rectifying the disorder, it is put back into the patient. So the patient is donor as well as recipient. So that's autologous. Allogenic means, so let's say there is somebody donates, like human embryonic stem cells or cord blood cells, right? So cord blood cells are taken and they are grown to see whether they are normal, and then it is injected back to the diseased person. So this cord blood cells are coming from a different individual. So there could be similarity because both are humans. There could be genetic similarity, but still it is coming from a different individual. So this becomes allogenic. So one kind of somatic cell therapy is autologous. The second kind is allogenic. Now again, there can be two kinds of approaches here. One kind is called as ex vivo and the other is called as in vivo. In ex vivo condition, the patient cells are taken outside, grown in the petri dish, right? Treated, and then it comes back to the patient. So the cells of the patient spends time in the lab. So this is called as ex vivo treatment. And in vivo treatment is when the gene is directly injected into the patient. In vivo means directly injected into the patients to treat the disease. Ex vivo is the cells are taken out, grown in the lab, and the mutation is corrected. Right? So similarly, it has to be done in the germ cells as well. The germ cells has to be taken out. It has to be treated in the lab condition, and in vitro fertilization has to be done in the lab, and then the embryo can be given back to the mother. So that's and the defect could be from the mother side, could be from the father side, and the respective gametes are corrected for the mutation, and the correct embryo that is formed after fertilization of the right gamete is again taken back inside the mother's body. As simple as it sounds, it is very, very challenging to correct these kinds of disorders. Now, the biggest challenge is how to, the biggest challenge for gene therapy comes from how to put the DNA inside the cells, how to shuttle the DNA inside the cells. Because if the right DNA doesn't go, how can it replace the wrong DNA, right? So there are broadly two types of processes by which the DNA is injected into the cells. One is through physical method and the other is chemical method. Both are not using the virus. Both are non-viral methods. In physical method, the DNA could be injected or micro-injected. It could be also be electroporated. What does electroporation means? Electroporation is a technique by which under the influence of electricity, the foreign DNA is inserted into the target cell. Right? Chemical method is that using various chemicals that can make the cell membrane more permeable, the foreign DNA is shuttled from outside to DNA using calcium phosphate, dextran, sulfate, or liposomes. So these are chemical methods to insert foreign gene. These are physical methods to insert foreign gene. In addition to this, these physical and chemical method, nowadays most popular methods are to use viruses. Viruses, it can with very ease enter into human cells. But we don't want viruses that can cause disease. So what scientists have done, scientists have genetically manipulated the viruses such that the bad elements from the virus genome has been removed and the virus cannot cause disease. So I repeat, these viruses which are being used as an agent to insert foreign DNA, in these viruses, the bad genes has been removed by genetic engineering. And only genes left are those genes which will help the viruses to attack or to insert the foreign DNA into the target cells. So some of these viruses are retroviruses, adenoviruses, herpes simplex viruses, and adeno-associated viruses. There are several viruses listed here. But the most popular or most reliable are retro or lentivirus and adenovirus. So this slide shows that the retroviruses are commonly used vectors or agents to shuttle the correct DNA into the disease cells to correct the disease. So as I mentioned to you, this figure, this is a very commonly seen figure. Here there is adenovirus. This adenovirus is made up of good genes and bad genes. The bad genes are removed and the good gene that would correct the disease is inserted through genetic engineering, mainly by restriction enzymes and DNA ligase. And this corrected virus is reformulated and then injected into the disease cells. And when this virus enters the cells, it injects, and this right DNA can be expressed inside the cells and it can cure the disease. Retroviruses have two steps, right? Retroviruses are those viruses which have RNA as their genetic material. And when they make DNA, it requires the enzyme reverse transcriptase that we have studied earlier. So retrovirus has two steps. First, convert RNA into DNA or cDNA to be more specific complementary DNA. And this complementary DNA has to integrate into the human cells. Now, what is the advantage between using adenovirus and retrovirus? So adenovirus can also take the new gene or the corrected gene inside the human cells. But retrovirus, it has the capacity to integrate into human chromosomes. So if it is the right target, if it inserts, the retrovirus has the capacity to integrate into the host DNA, right? Through what enzyme? Through integrase. So these retroviruses have two unique enzymes. Number one, reverse transcriptase that can convert RNA to cDNA. Second, integrase. Integrase is an enzyme that can integrate the cDNA into human chromosomes. So if the correct DNA is integrated at the right place, it can be a permanent solution because the corrected DNA is part of human genome. So, but these steps are very challenging in terms of manipulating these viruses, right? And then more challenging is to insert into the right cells. And even if you get the right cells, it has to go and integrate into the chromosome. Because if it integrates into a wrong place, it can give rise to secondary mutations. So on one hand, these technologies, these new technologies, they're very high risk, but it is doable because of various advancements. What are liposomes? Liposomes is an example of chemical gene therapy. Right? Liposomes is essentially spherical body made up of lipids. Now these lipid molecule, it's very interesting. These lipid molecule could mimic the cell membrane of, let's say, lung cell. So let's say somebody has leukemia, and this leukemia emerges from bone marrow. So the bone marrow cells can be taken out from the patient, can be cultured, nourished in the lab, and the new gene can be inserted into bone marrow using liposomes. So what are liposomes? Liposomes are lipid vesicles, lipid spheres. These lipid molecules can easily integrate into the bilipid layer of the cell membrane. So once it integrates, then the DNA is delivered. But the challenge here is that with the liposomes is to how to make liposomes, which is very exactly similar to lungs or to bone marrow membrane. That is the challenging part. How to generate liposomes which will exactly resemble the membrane of the bone marrow. So it's very tough to insert foreign DNA into our cells. Scientists are also trying various methods like inserting naked plasmid DNA directly into the cells. And you can argue, or you can ask me a question whether this Moderna vaccine or Pfizer vaccine, or even vaccine that is, Pfizer and Moderna vaccines, which are based on RNA, is it a gene therapy or it's a like the ultimate goal is to make spike protein? So overall, I can say it is both because we are using mRNA, right? The classical vaccine would be, for example, the polio, like they make polio proteins, and that polio proteins are injected into our system, and then to generate antibodies to counter, polio virus. But here we're injecting oligonucleotides, which are mRNA. So these new strategies are somewhere in between. They're using nucleotides, so of course they're using gene. But gene is not human. This gene belongs to COVID-19. But ultimately, it is what we are getting as a vaccine is mRNA for spike protein. So it could be a type of gene therapy, but may not be to treat mutations, right? It's a strategy to develop antibody to combat infection, combat a pandemic, right? So think about it, what I am mentioning, and you could share whether this new vaccine strategy using oligonucleotides, mRNA, RNA, is it in between classical vaccine or between gene therapy? So think about it. Many new discoveries, innovations are coming that will change the old way of thinking, the classical way of thinking. That's the point I wanted to convey to you. P53 is a very unique target. Why it's a unique target? Because this protein is mutated in 70% of the cancers. Right? P53 is a tumor suppressor gene, and it can control cells. It can control the growth of the cells by causing cell cycle arrest. Or it can trigger apoptosis to kill the cancer cells. But if P53 is mutated, then neither the cells will be arrested nor apoptosis will be mediated. And in 70% of the cancer, P53 is mutated. So if we have a gene therapy approach to correct the mutation within P53, and this P53 can do the function of tumor suppressor, it will be an enormous, enormous relief to various patients who are similarly in cardiovascular diseases, right? It's cystic fibrosis. There are endless number of genes which undergo mutation and causes debilitating diseases. For example, Huntington disease that causes a skeletal bone system, cystic fibrosis. And there are companies now, Novartis is coming with the treatment of Huntington or cystic fibrosis. There are various gene therapy strategies are in clinical trials. So, it's a matter of days when this will be available. So, so ocular pathologies, some people lose their eyesight suddenly. And so people figure out that there could be a mutation. So those cells in the eye could be treated with gene therapy, like macular degeneration. So, gene therapy, the concept of gene therapy was in late 70s, but it is very powerful. And at the same time, it is very challenging. Challenging in what aspects? Let's count. Number one, to design a good DNA that can integrate into the cells. But before it integrates into the cell, it has to go through the cell membrane. So entry into the cell, integration into the cellular nuclear component so that this new DNA can transcribe and translate. And of course, a good vector system like lentivirus or adenovirus. So it has to be, it takes these experiments that we're talking in just one class. It takes years to do that. And work of those people, those scientists, they come to the classroom as textbooks, right? So, but technology, information is growing in leaps and bounds, right? And it's at this stage we have to keep up with the technologies because we have our future, right? So with this concept and with this chapter, I finish this chapter on gene therapy. I'll put this video upload this video on the Blackboard after a few hours. And there's an assignment on the Blackboard. So thank you very much. It's a pleasure. Bye. Okay. Good night, Naomi. Take care.